This is the first lesson of chapter 24, where we're going to be talking about early life on the Earth and the diversification of one group of organisms, the prokaryotes. As always, these are the learning outcomes that we're going to be focusing on in this chapter. Some of these we'll touch on in this lesson, but the rest will be in subsequent lessons that may be in videos and/or in class. And during the lesson, there might be some videos that I have shown here on the slides. Unfortunately, I won't be able to play them in my recorded video because you won't get the audio. So this is a good opportunity to, when you see these, to go ahead and pause my video, go to the website shown here, which also I have the shortcut link in D2L in the chapter 24 module, and take a couple minutes to watch the video about how we can relate Earth's history to a football field. And I have another variation of this activity that we might do in class, but not with football field. So if you want to go ahead and do that now, pause this, check that video out, and come back here. Another way that Earth's timeline can be depicted is through this really unique piece of art designed by an artist, Pablo Carlos Budassi. I really like it. Not only is it kind of visually appealing, it looks like a shell, a spiral shell of a mollusk, but it really packs a lot of information into a really small space. And of course, you can't see all the little details unless you zoom in, which I encourage you to do that if you want to go to the website here, and you can zoom in and you won't lose any resolution of that image. And this actually goes back to the beginning of the universe. So I want to focus on a couple parts of this time spiral so that we can really start digging into Earth's early history and the origin of life on Earth. So maybe you can see early Earth formation here on this, even though it's very small to read. We're talking about this ball of magma, basically, that's cooling and hardening and starting to form our planet. So that's the part I want to take a moment to look at in a little more detail, and now you can see some of the relevant text. It was about 4.6 billion years ago that Earth is thought to have started forming. And so if we look along that part of the spiral from left to right, you can see some of the key events leading up to the earliest life forms. The earliest known life forms based on fossils that exist today are the stromatolites, which are thought to have started existing 3.5 billion years ago. And we will learn more about this group of organisms because there are actually stromatolites alive today. But the fossil remains that exist can be dated back 3.5 billion years ago. And so what stromatolites are, just briefly, they are prokaryotic and photosynthetic. So they are a group known as cyanobacteria that have the ability to photosynthesize, make their own food from sunlight, but they are single-celled prokaryotes that lack a nucleus. And this group is also thought to have been responsible for putting oxygen into Earth's early atmosphere, which paved the way for subsequent groups of organisms to evolve that required that oxygen to have aerobic cellular respiration occur. So now I want to back up a little bit into this middle area here where you see some other types of cells, and we don't have the fossil evidence for these. We only have the first fossil evidence of the stromatolites 3.5 billion years ago, but of course it's thought that the stromatolites would have evolved from earlier, more primitive life forms, and we only have hypotheses for how this occurred, for how Earth went from having non-living, no life, to having the molecules and the ingredients necessary for life to start existing. So we're now going to be looking at the hypotheses for how the first simple cells came to be on Earth. These are the four hypotheses in order of the stages that are thought to have occurred leading to the first simple cells. So just briefly, they're here, but we're going to go into the more details of these, including some of the scientists and experiments that specifically led to these hypotheses. So let's start with the first one, the synthesis of small organic molecules. What we know about this part is first attributed to the work by Oparin and Haldane in the 1920s. Oparin was a Russian chemist and Haldane a British scientist, and they independently were working on a similar hypothesis about Earth's early atmosphere. So they weren't working together as a team, they were in different parts of the world studying these things separately from one another. And that's kind of cool to think that two people had a similar idea but never had talked to each other about that. Of course, information was a lot harder to disseminate across the globe back in the 1920s. There was no internet. And yeah, so they weren't really calling each other on the phone to talk about this stuff. In any case, they both hypothesized that Earth's early atmosphere was a reducing environment. And if you remember, reducing means has to do something with electrons. Reduction is gain. Reduction means gain of electrons. So the atmosphere had a lot of electrons available to add to compounds that were looking to gain electrons. So this is part of the necessary conditions for these small organic molecules to form in that reducing environment. So the experiment jumping ahead, never mind, not about the experiment here. So they believed that the organic molecules would be synthesized using energy from lightning and ultraviolet radiation, again, related to the fact that the environment was a reducing environment. And the Oparin-Haldane hypothesis is also consistent with what Darwin hypothesized based on a letter that he wrote to a friend in 1871. You can read the whole quote here at the bottom right. But basically, he was thinking the same thing, that if the conditions were just right and all the ingredients that were there, inorganic molecules could chemically change to form organic molecules. So I, in red text, kind of emphasized a few things that I thought were really important about what Darwin was saying, with the presence of ammonia and phosphoric salts, light, heat, electricity, that a protein compound could be chemically formed, which again, organic molecules include proteins, and this is the first step needed in the transition to the first cells, the first living cells. And Darwin called this this condition, and the fact that Earth at the early stages was really mostly covered in water, and it's thought that the oceans were the place where life began. So he called the early oceans primitive soup or primordial soup. So you'll sometimes see artistic renditions with Darwin and the reference to primordial soup, and it's basically the idea that there's organic molecules present in Earth's early oceans that pave the way for cells, for life to form. Okay, now we're going to fast forward a little bit in time, still corroborating that first step, the synthesis of small organic molecules, an experiment conducted by Miller and Urey at the University of Chicago. They worked together on this to simulate the conditions of Earth's early atmosphere in the lab and show how when those ingredients are present, including methane, CH4, ammonia, NH3, hydrogen gas, water vapor, and electricity simulating lightning, and again, that reducing environment, that they were able to create in the flask here, collecting the cooled condensated water with organic compounds, the first organic compounds. So they simulated this, what they think would have happened in the early Earth, and they were able to replicate that in the lab. And several subsequent experiments like Miller and Urey's to test that hypothesis showed the same thing. It's still being tested today, but the consensus is out there that non-living or inorganic molecules can be made into organic if the conditions are right. So I also found a video that I like about the Miller-Urey experiments. And again, you'll have to go to the YouTube link here, which is also in D2L to watch that to get the audio. So if you want to go ahead and do that now, pause my video, go check that one out, and then come back. Okay, now let's move on to step 2, which is how the small organic molecules that we just saw how they could have been formed would join together to form larger molecules or macromolecules. And we're really focusing on RNA as the macromolecule, ribonucleic acid, because it has been shown that this hereditary molecule of RNA can be produced spontaneously from simpler molecules. And this can be done without enzymes or ribosomes that would have come later in the progression of the simpler molecules to the more complex and ultimately the cell. So first would have been RNA monomers, and then later on the other more complex molecules like enzymes and ribosomes. So now we've seen how inorganic molecules can be then made into organic, and those small building blocks of organic molecules can be used to build RNA, which is a hereditary molecule that contains genetic information. So the next step is hypothesized to be that the RNA molecules would be packed into a protocell. A protocell is a fluid-filled vesicle with a membrane-like structure. Think of proto meaning like just before cells. So this isn't really the cell just yet. It's the thing that came before the cell. It has that membrane to separate it from the external environment. And as we should remember from Biology 1, lipids, which lipids that make up the cell membrane, can self-assemble to form a layer or a bilayer around other components, causing there to be that membrane there. So we know that that can happen without a living organism orchestrating that, just the nature of those molecules, how they assemble when they're near each other. But some scientists have discovered that another molecule or component might have comprised the outer layer of the protocell, and that would be actually inorganic particles of clay, and that's shown at the bottom right here. This is from an article that I found from Harvard. You can check out that link there if you want to read more about it. Scientists have discovered clay armored bubbles that exist today that may have formed the first protocells. So that's a very interesting and new development in the field of science that could explain how those protocells formed when life wasn't yet present. All right, so and then now we come to the final stage, step 4, which is the fact that the RNA that was now contained inside of these protocells has the ability to self-replicate. It is thought that the first genetic material was probably RNA and not DNA because of RNA's ability to replicate itself without any additional machinery needed in that protocell. So there are these things called ribozymes, which are ribonucleic acid enzymes that are responsible for the copying themselves process. And as you can see here, RNA comes in a diverse assortment of three-dimensional shapes. And the shapes that would be more easily replicable would be the ones that would be more favored in the descendant RNA molecules that would ultimately be the early hereditary information of the first cell. So in conclusion for this first lesson, that brings us to this idea that there was, before life really took off, an RNA world of small RNA molecules that were able to replicate, that were able to store genetic information, and that were carried by vesicles. So check out this video about the RNA world hypothesis before we go on to our next lesson.